And good morning, all you friends out there in YouTube land. So, we've been discussing basic systems in the cell, covering, most recently, the idea of protein processing and protein localization. The idea being that if an RNA contains the nucleotides that will in turn code for a signal sequence, once that signal sequence gets translated, because everything starts out being translated on free ribosomes, once that signal sequence gets translated, that protein will be picked up and bound by the SRP and escorted to the ER. And that's how ER becomes rough ER, is that it has ribosomes sitting on top of it. The ribosome then continues translating the protein, and as it's translating it, it's dumping it into the lumen of the ER. This is the co-translational translocation mechanism. And just in our little drawing here, we have a protein that, other than the signal sequence, doesn't have any big hydrophobic stretches. And so it gets dumped into the lumen, and signal peptidase clips the amino terminus part where the signal is, and this thing is floating free. It now gets taken by transition vesicles into the cis space of the Golgi and makes its way through the Golgi into the trans space. And as it's being processed through, there's protein processing, there's protein folding, there can be proteolytic modifications, and perhaps most importantly, there's glycosylation, the addition of sugar molecules to specifically like asparagines. And these sugar molecules can be fairly large polymers. They're then further modified in the Golgi. And at the end, the protein is going to be in the stack of the Golgi that is at the side closest to the cell membrane, and this is the trans space. And there is a whole network of essentially protein railroad tracks made of microtubules that are escorting the vesicles to the cell surface where they fuse, and this stuff gets dumped out. And so we now have a protein being dumped out into the extracellular space. At the same time, there are vesicles being pulled in, and some of this is just simple vesicles being pulled in and maintaining the membrane integrity. Sometimes you can be bringing in different kinds of molecules. There's a whole notion and a Nobel Prize winning idea of what's called receptor-mediated endocytosis, in which a molecule binds to its receptor, and that whole thing is then brought into the cell. And this is how cholesterol is taken up. And we then end up having vesicles on the inward flight that get turned into lysosomes and peroxisomes. These are sort of the hydrolytic spots in the cell where things that are being brought in, including components of the cell membrane, can be recycled and then put back out into the system. So that is the easiest story in terms of a secreted protein. If we've got a protein that's going to be bound to the membrane, which we're about to get into, something like a receptor, then there's going to be a transmembrane stretch, or generally going to be multiple transmembrane stretches. In the simplest model, with a single transmembrane stretch, the signal peptidase clips the amino terminus, and so the amino terminus is floating free inside the vesicle. The carboxy end is still in the cytoplasm. And so a way to think about this is carboxy with a C in cytoplasm. So the carboxy end is going to be facing the cytoplasm. The amino terminus then is inside. When that fuses with the membrane, bada bing, we have the amino terminus facing out, the carboxy terminus facing in. Another thing that happens is there is essentially no glycosylation going on in the cytoplasm. So only the portion of the protein that is in the vesicle and exposed to the modifying enzymes in the Golgi can be glycosylated. And so we end up with a very distinct sort of configuration of a cytoplasmic end and then an extracellular end. So receptors have to be placed into this system in the appropriate context, and that you have to have the binding side of the receptor facing to the outside. And then inside, we're going to have this next set of things happening we're going to be talking about. So that is how we get proteins to the cell surface. Now, the next step in thinking about cell organization is talking about cell signaling. This is the mechanism by which cells can communicate with each other. And as I was walking across the street thinking about this, single cells don't need to do a whole lot of cell communication. And that steps us actually into a couple of interesting areas, and that there are some bacteria, that particularly those that are motile, they can swim around like little bacilli, they really spread on the plate. They actually are sending signals to each other. And there's a whole study and a story of what's known as quorum sensing, where if a bacterium sees food, it can begin sending out chemical signals that will cause everybody else to begin clustering around it. And so single cells do a bit of this. If you're a non-motile single cell, yeah, you're talking to yourself. You don't really send out secreted signals. It just doesn't do you any good. In multicellular organisms such as us, this is a critical part of the mechanisms by which multicellular organisms are able to carry out their jobs. It's how cells are able to differentiate, how a cell knows where it is within an organ. It is also the mechanism of hormones. And so hormones we can define as in a couple of different ways. There is standard hormonal signaling, in which you've got a tissue, say in the anterior pituitary, someplace in the brain, your adrenal glands, that are secreting a soluble molecule that's in the bloodstream. And then there are target cells that respond to this. So hormonal signaling then is going to be generally a small signaling molecule, a hormone, and it's secreted by one tissue in one place, and then we have the responding cells. The responding cells, in order to respond, they have to have the appropriate receptor. And so we can have hormones floating around in the milieu of a large organism. If the cells don't have the receptor, it's like the hormone doesn't exist. If the cells have the receptor, they can bind the hormone and carry out some kind of cellular mechanics. So, if you don't have the receptor for estrogen, taking birth control pills, like a dude, isn't going to do anything to you. Only if the cell has a receptor can it respond to a hormone. So we have kind of a two-pronged way of operating the signaling system. Is you have to send the signal, and there are certain cells that will then send the signal. If the signal doesn't go out, nobody cares. And then only if you have a receptor can you respond to that signal. So there are hormone systems in which you've got the hormone being secreted by a portion of the nervous system up here that is then responding to cells all around. The inflammation process is one example where you have a little boo-boo on your finger and there are neurons surrounding it and then other parts of the body that are secreting signals in response to that inflammation and that damage that then cause that part where you have your little boo-boo to begin aching. It doesn't happen someplace else because the signal is being targeted to this spot. We also can have sort of a autocrine system that's actually the right term, regardless, where you have a single cell that secretes its own hormone, and then there is a cell right next to it that will respond. Or a cell can actually respond to its own signaling system. The immune system does this pretty well. If you have an antibody-producing cell that binds to an antigen, so when you get your vaccine, there are just a few cells in the body that are presenting an antibody that can bind to the vaccine. When they bind, they are able to signal themselves to begin dividing more. And so that's why it takes about 2 weeks or so for this cell division to occur to accumulate enough antibody-producing cells that you now have immunity. So there's all kinds of hormonal systems. Your brain is carrying out tons of cell signaling events. This is how, really, when you're trying to access memories, you're engaging in different kinds of signaling events and things. All right. So when we begin thinking about hormones and receptors, we can begin breaking things down into a couple of different requirements and mechanisms. Think about this. The book has a somewhat dated picture of a cell phone, in which the cell phone is sending out signals and is also able to receive signals. You know, that's what we're talking about. Now, if we break these signals down, we can break them down into two very broad classes. And what we're going to be doing is building an organization chart of such and breaking cell signaling down into these different kinds of classes. And then with each class, we'll be doing a little bit of discussion. But we can have our very initial breakdown based on this. Is the hormone is going to be nonpolar and therefore diffusible across the plasma membrane. And we've been talking a lot about the membrane impermeability to a variety of different things. And so we know that if a molecule has got a charge on it and is bigger than a molecule of water, or if it has a strong enough charge like a chloride or a sodium atom, there's no way it can get past that hydrophobic phospholipid bilayer. So nonpolar molecules, things that are based on a large carbon structure that don't have any polarity, kind of oily type guys, they can diffuse across the plasma membrane. And so I were to say to you, what might be an example of a nonpolar hormone? Say something based on cholesterol, a sterol. We might think to ourselves, how about a steroid? And I'd say, great example. And so a perfect example of a nonpolar diffusible molecule, something that can pass across the cell membrane, are those hormones based on cholesterol and things like steroid hormones. Now, these are the most obvious class of nonpolar hormones. If we were to think about it, there are a range of other nonpolar hormones, but this is where we're going to hit. This is a good one. And so one of the first things we can ask is the mechanism of steroid hormones. And so we'll get into the polar non-diffusible guys in just a moment, but let's just take a moment here to talk a little bit about steroid hormone mechanism. And we'll kind of be hitting back and forth on this. So steroid hormones, things like testosterone, estrogen, a range of different kinds of stress hormones, cortisol, things like this. They have the ability, there's an O there. They have the ability to diffuse across the plasma membrane. Consequently, what we can say about this class of hormones is they're going to have a cytoplasmic receptor. So the receptor for the steroid hormone molecule is actually floating around the cytoplasm. And generally they're kind of anchored in the cytoplasm, waiting for a steroid hormone to diffuse across. They bind, and now they can do something. And what that something is, and this is pretty much condensing a lot of stuff, we'll talk a little bit about some of the experiments, is that once the steroid hormone receptor binds the steroid hormone, it then is going to move into the nucleus, and there it's going to bind DNA and act as a transcription factor. It's going to bind something that is known in a generic term as a steroid response element, aka an enhancer. and we've talked about enhancers. These are things that are distance and orientation independent. These are DNA sequences that serve as a binding site for a transcription factor. The steroid hormone receptor is, in fact, a transcription factor. It's just waiting for that hormone to bind so it can then get into the nucleus and actually act as a transcription factor. So we've got then this link here to how a steroid hormone acts at the level of gene expression. Steroid hormones are going to turn genes on. An absolutely classic experiment, again with fruit flies, and one of the major contributions that flies have made to our understanding of science is an experiment done by a guy named Mike Ashburner back probably in the 60s or early 70s, in which he was using the steroid hormone that's involved in triggering molting, what is known as molting hormone, a ecdysone. And it's a classic, based on cholesterol, steroid hormone. If you inject that into a cell system, particularly salivary gland cells, they've got really big chromosomes. And the injection of, or the addition of ecdysone, the steroid molting hormone, just placed on top of the cells, it absorbs, diffuses in, and within a few minutes, you begin to see changes in the chromosome. One of the things that happens with fly chromosomes in the salivary gland is that they are giant, look like ropes, because they undergo round after round of DNA replication without actually undergoing karyokinesis, where they don't separate out. And so they're just stacked on top of each other, all these different rounds of DNA. And you get areas that are engaged in a lot of transcription that begin to puff out, and these are known as puffs. So people had observed that during molting, there are a set of puffs that come out. And what Ashburner was able to demonstrate was the addition of the steroid molting hormone actually causes the puffs to puff out. Now, one of the cool things that's actually a really interesting experimental model is that if you let it go for a couple of hours, you see a whole range of puffs. And what he then did was added ecdysone along with a protein synthesis inhibitor. A protein called, or a drug called cyclohexamide would do it really well. And what he sees is that the puffs still begin to form, but not all of them. And as he begins to dissect it, he can begin to see that there are some puffs that are early, and then there are some puffs that are late. They take a couple of hours in order to come up. The late puffs are actually responding to transcription factors that the early puffs are generating. And by blocking protein synthesis, he's able to have the initial puffs come up. That's where the steroid hormone receptor is binding, acting as a transcription factor, turning those genes on. But then the next set of puffs is requiring the actual gene products to be translated, turned into transcription factors, migrate back into the nucleus, and begin acting on other sites in the genome. So what we can demonstrate from this is that steroid hormones act at the level of gene expression. Important enough that we'll write that here on the board. So steroid hormones act at the level of gene expression. So the steroid hormone receptor is a transcription factor just waiting to be able to get escorted into the nucleus to do its job. Now, as we have spoken on a number of occasions, to go from a quiet gene to one that is being transcriptionally active doesn't take very long. You have the transcription factor binding. RNA polymerase is recruited. You begin making RNA. That RNA now has to be processed. It has to be escorted out of the nucleus into the cytoplasm. It has to be translated, and very often the translation product is going to get into the Golgi. And so at best possible speed, you're talking an hour and a half or two, probably 3 hours, for sufficient amounts of protein to accumulate. And so if you were to, in deep meditation, tell one of your cells to begin transcribing a gene right now, and it starts transcribing it, it's probably going to be getting close to lunchtime before you have sufficient protein that you can actually say, okay, I've turned this gene on. So it is not a fast process. It is a very slow process. But because we are actually changing gene expression, it is going to be a fairly long-lived process. And so that's another key point to make with steroid hormones. And as you'll see, we'll be contrasting that in just a moment. So steroid hormones and pretty much all of the nonpolar, diffusible across the membrane type hormones, they're acting at the level of gene expression. And consequently, they are going to be slow on the order of, just for the number, 3 hours, 2 to 3 hours, but the effect is going to be very long-lived, as long as that protein's hanging around, which could be weeks. So it is slow, but it is very long-lived effect. So that is the mechanism of a specific class of hormones, the steroid hormone class, and pretty much any kind of diffusible across the plasma membrane class. Again, as I was writing this, it was like, probably have to do a deep dive into who knows where to think about, are there diffusible hormones that actually aren't acting at the level of gene expression? And there really aren't. Off the top of my head, I can't think of anything. So I'm sticking with this. As a general rule, this is the case. Now, what about hormones that are polar? That is, they can't get across the plasma membrane. Glad you asked. So now, what we got here is the beginning of our chart, in which at the very top it says hormone signaling, and then we bifurcate and we have diffusible across the plasma membrane, i.e. steroid hormones. And then on the other side, we're going to have the hormones that can't diffuse across the plasma membrane, and I'll tell you that this is the side that really gets a lot more complicated. This side's kind of hanging up here beep, by itself, fairly simple. This next side, oh yeah, we got lots of bifurcation points and all kinds of things happening here. So, we have now the non-diffusible class. Non-diffusible hormones, and there's a bunch of these. And these are things that are often, very often, they are small peptides. And that means they are either a few amino acids in length, or maybe 25 or 30 amino acids, 50 amino acids. Insulin is a great example of a peptide hormone. Now, again, we're kind of trying to keep things in a fairly simple model. There are other molecules that are non-diffusible that aren't based on amino acids. There are some different kinds. Some of these things are even gases and things. But for the most part, let's just go ahead and talk about peptide hormones. I like that. There are certainly individual amino acids that act as different kinds of hormones. The neurotransmitters actually can be placed into this class of cell signaling. But let's stick with small peptides. All right. So now, with the small peptide class, or the non-diffusible class, in order for this to work, we're going to need a receptor that is sticking out into the extracellular space. We're going to need a transmembrane protein that is acting as the receptor. Because these molecules cannot diffuse across the plasma membrane. Without that receptor, it is like it never even happened. It just bumps along, nobody's home, there's no key for this lock. There's no doorbell for this finger to ring, whatever. All right. So, under the heading of peptide hormones, non-diffusible hormones, we now are going to bifurcate once again. We've got a class of receptors that are known as receptor tyrosine kinases. They are, of course, abbreviated as the old RTKs. Receptor tyrosine kinases. And as it turns out, insulin binds to a member of this category of receptor. A receptor tyrosine kinase does this. We got the cell membrane here, the plasma membrane. The receptors are usually two little equivalent pieces, or two proteins, rather than one integral protein. And in the case of insulin, and probably with most of these kinds of receptor tyrosine kinases, insulin actually works as a dimer. And when insulin binds to its receptor, it causes a change in the shape of the internal portion. Now, do we remember what a kinase is? Ooh, hopefully you do. A kinase, the most critical enzyme class, they are going to phosphorylate, add a phosphate, a substrate. And it's going to be another protein. Just as a little nerd point, when we talk about phosphates, and remember what you were thinking as soon as I said phosphate? Massive negative charge. Thank you, Catherine. When the phosphate is attached covalently to a side chain of amino acid, we actually can see that in this kind of signaling event, it is generally the amino acid tyrosine. And so tyrosine phosphorylation is involved in cell signaling. When we're talking about phosphorylation involved in protein changing shape, like an enzyme, like the sodium potassium ATPase, or as we start talking later, muscle contractions and things like this, these are amino acid residues of either serine or threonine. And so we can kind of define kinases into these two classes. Tyrosine kinases, those are big-time cell signaling systems. Non-tyrosine kinases, they are involved in transferring energy, but they're not doing cell signaling. So the kinase is actually integrated in the receptor. And what the receptor is going to do, it's going to cross-phosphorylate itself. It's going to add a phosphate over here. And this guy is going to phosphorylate this guy. And they actually do it several spots. So you got these big negative charges here floating around. And so they are cross-phosphorylating each other. And what this phosphorylation does is essentially set up like a little bit of a whistle or a docking spot, that there are a whole class of cell signaling proteins that then get recruited and activated. And just as an example, but not exhaustive, there is a protein known as MAP, or mitogen activated protein kinase. Another protein that we'll be looking at in a bit is a protein known as RAS, which is the abbreviation of the Rous avian sarcoma virus. It turns out, and we'll be getting into this in 2 weeks, mechanism of cancer, a ton of cancer-causing proteins are involved in the next round of cell signaling. When we start talking about cancer, just as throwing this out, because RAS and MAP are both oncogenes, these cancer-causing genes are present in us all the time. They're critical to our operation. And they are only cancer-causing when they are mutated in some way. That's why we would talk about the fact that any mutagen is a potential carcinogen. So under normal circumstances, RAS and MAP, they're behaving perfectly well and they're very well controlled. Under abnormal circumstances, RAS can be mutated in a way that it acts like it's being signaled all the time, because the next thing these guys do is phosphorylate or activate downstream proteins that are then having all kinds of fairly quick effects. So there's the MAP kinase, and then there's the MAP kinase, and I think there's even a MAP kinase. It has to do with sort of developing terms, and you're going, oops, we probably should have called it something better, because there is a kinase that phosphorylates another kinase that in turn phosphorylates another kinase. You have this big kinase cascade, which we're about to get into. So the receptor tyrosine kinases, they are their own little one-man band. They are cross-phosphorylating. How do you do? Boop, big phosphate sticking out. Begin recruiting all these proteins that are then activated, and they in turn are causing changes in the cell metabolism. Cell division can be triggered this way pretty well. So that was the receptor tyrosine kinases. Now, as we continue on with peptide hormones, we're going to bifurcate, or this is kind of at the top here, I guess. And then we have another class of peptide hormone response systems. So there were the receptor tyrosine kinases, and now we have a class of receptors that are known as G protein coupled receptors. And of course abbreviated as the old GPCRs. So G protein coupled receptors, they are yet another really big deal. The receptors themselves actually can be identified in the genome because they all share a significant amount of amino acid homology. They tend to be 7 membrane pass proteins, that is, fairly complicated organization that they're passing through with multiple starts and stops in the membrane. And they are interacting with a G protein. So, the G protein coupled receptors. And the experimentation to identify these took quite a while. Here is a really useful way to think about G protein coupled receptors in a bit of a generic terms that the book doesn't really use, but I like a lot. We've got the receptor. It's transmembrane protein, stick in there. It is going to be in the membrane physically close to a G protein. A G protein is in fact a generic kind of protein. There's really just a standard gene set that produces a G protein. It's got a couple of different subunits that we'll look at later, but we're not going to worry about that right now. The G protein is sitting right here. It is cytoplasmic. And the G protein, like the name implies, it is a GTP binding protein. In its inactive form, it is not binding GTP. In the active form, it binds GTP. All right, the G protein is right next to the receptor. When the receptor binds its ligand, the hormone, the G protein is tickled and it splits apart. And a portion of the G protein is now going to come over here and interact with another box that we will call the effector. The effector is actually doing the actual cytoplasmic signaling. The effector is going to produce something that is called the second message. This is the internal message. It's actually known in the biz as a second messenger system. I mean, that's what this used to be kind of referred to as. So the first message is the hormone itself, but it hangs out here. It can't get into the cell. The second message is that which is being generated inside. And so that's why this is known as a second messenger system. So this is a little historical point. There is a classic phenomena have been observed with, I think, well, for the story we're going to go with liver cells. When you hit liver cells with adrenaline, they begin producing large amounts of glucose. The thing gets dumped into the blood. And as I'm thinking about this, the original experiment wasn't using this, but it's all right. And it happens really fast. So if you think about this from a physiological perspective, you are a small creature or a primitive hominid, and you're standing around in the forest as a little mouse, you're wandering around, and you come across some kind of great big predator. A large saber-toothed cat jumps into your path. And you look at that saber-toothed cat and you say, "Good morning, good sir.
Goodbye."
You look really frightening to me. You know what I need? I need a big burst of glucose coursing through my bloodstream to power my muscles, because really what I want to do is run like hell and climb a tree. Now, I'm going to begin digesting my food, extracting glucose from it. I'm going to begin breaking down, because I have glucose storage products in my liver in the form of a starch-type molecule that we call glycogen. I'll begin breaking that down. Give me a few moments. I have to turn a couple of genes on. So really, you have a seat, I'm going to have a seat. 2.5 hours from now, man, I'm going to be ready to run. Okay, the saber-toothed cat just quietly saunters up to you and begins chewing your leg. This isn't going to work. We need to have something that is going to happen fast. So we can't depend on a cell signaling system to actually act as a transcription factor all the time. Because I need to have a result in the next 10 seconds or we're going to be eaten. Adrenaline is an excellent example. How many of you have ever had a burst of adrenaline, some kind of fright? A classic thing you can actually do is tell the class when they walk in, okay, so we're having our exam today, and everybody's like, and you have potentially had this experience of where you feel this pit in your stomach, you get very jittery, it's like, holy smokes. In our modern world, we have adrenaline responses that probably are not really very useful, because it's not a matter of running, it's a matter of going, oh shit, keep your hands on the steering wheel. Yeah. Or just pull over quietly and sit there. And regardless, I used to often think,
system.
yes. Doing my drive back and forth to Bloomington, if I were to see the policeman as I drive by and I'm going a little bit over the speed limit, it's like, oh no. And then, in the event that you see flashing red lights behind you, the response is, I'm either driving to Alaska, I'm driving to Mexico, I'm going to floor it, that's it. So the adrenaline response in the modern era doesn't always serve us, but we'll get into that. So you have this really, really fast response. Now, what these guys were doing in the very early stages of figuring this out is, all right, we know that adrenaline has this very specific effect on liver cells. What if we take adrenaline and taking a teeny needle, and they actually did this, and inject it into an individual cell and see what happens? Guess what? Nothing happens. Because the first messenger requires its cell surface receptor. Adrenaline actually being injected into the cell, there's no receptor there, it doesn't do anything, it's useless. Now, if you were to do that with steroid hormones, it would just be that much faster. It's like, ooh, steroid hormones work by injecting into the cytoplasm. But peptide hormones, no, nothing happens. what they began to observe was, in the case of adrenaline, there is a molecule that is very quickly coming up within a matter of seconds. And someone has to say, hey, that's the kind of speed we're talking about. It is a form of adenosine known as cyclic AMP. This is based on the ATP molecule. We're going to pop off two of the phosphates, it's a pyrophosphate, and it cyclizes. The chemical formula of it, if you really want to memorize that, is in the book. No, we do not need to know that. This is cyclic AMP, or CAMP. We actually mentioned cyclic AMP a little bit in bacterial gene expression when the catabolite activator protein is active, which it binds to a portion of the upstream region, allows RNA polymerase to bind a little bit better. It's actually responding to cyclic AMP. This is a classic, even with bacteria, stress response. When you get stressed out, you begin producing cyclic AMP. And the enzyme that does that, so what these guys discovered was, if you inject adrenaline into a liver cell, nothing happens. You inject cyclic AMP into a liver cell, kablooey, everything starts to happen. You begin producing tons of stuff. Cyclic AMP is, in fact, not the second messenger, but a really critical second messenger. So this is a second messenger. And the enzyme that generates cyclic AMP from ATP is an enzyme that actually fits into this little box as the effector. It is the enzyme known as adenylyl cyclase. Adenylyl cyclase is taking ATP and converting it into cyclic AMP. That is big time activation. And it does it really fast. And rather than getting into the next step of it, we're going to save that for Wednesday, but we'll talk a little bit about it. Adenylyl cyclase, what it's doing is generating large amounts of cyclic AMP. So let's put that here. We got ATP is being converted into cyclic AMP, and there are a whole class of proteins that are cyclic AMP binding proteins that are being activated. So this means rather than waiting for gene expression, we've got a whole set of proteins in the cytoplasm that are cyclic AMP dependent. We'll get into them on Wednesday. And when cyclic AMP is present, man, they just explode with activity. Woo! All right, so just as an example, in learning and memory, you can actually do some very clever experiments that have been done in flies where you might not think about it, but flies actually can learn and remember fairly simple things. They actually can be trained in kind of standard Pavlovian response training, in which you take flies and you put them into a chamber with a little bit of a metal grid on the bottom and hooked to a power supply. And you blow an odorant across them, and every 10 or 15 seconds for like 5 minutes, you give them a brief but significant electrical shock. And so they're sitting there smelling the smell of cherry lifesavers and getting an electrical shock. They don't like that so much. And then you evacuate the chamber, put them into a fresh chamber. Now you put in, a banana flavor, and they're going, and no shock. And they're like, You do this a few times, and guess what? Flies go, we like banana flavor, and we don't like cherry flavor. And normally, they're like, we like them both. But when you train them up, and then you can put them into a t-test where you have banana odorant going one way and cherry going the other. They don't actually use banana and cherry. They're using various other alcohols. But better than 90% of the flies will make the correct choice, and they'll head over to the banana flavor and leave the cherry flavor alone. Except that one guy going, I like it, but So you can train them, and they can learn this. I mean, it takes a little while, but they can learn it. But even cooler is the next day you put them into that tea chamber, and still a good percentage of them go over to the non-shock associated odor.
So they're remembering it.
They actually remember. And then over a series of days, they begin to forget. And eventually, after about 72 hours, about 3 days or so, you'll end up where it's 50-50. Some go this way, some go that way. There's no specific choice. They have forgotten. So this is an assay that a geneticist can use to look for mutations. And it is a very tedious assay when you think that most of the mutagenesis screens that I would always do would be based on either a wing shape or a pupil case shape, so you can hold the vial up to the light and go, nah, that's no good. Nah, that's no good. Because, you hit a mutant in your gene area maybe once every 1,000 crosses, so you got these 1,000 vials, then you just very quickly flipping over your shoulder. When you're doing the learning and memory, you got to go through and train every single one of these fly strains, these mutations that you've generated, to see, okay, are you doing this? I mean, one of the things you do is, can they smell? Because if they can't smell, then they're not going to be able to do the odor avoidance. Can they move? Because if you can't move, that doesn't count either. But you go through this, and this guy named Ron Davis, actually, and his crew at Baylor College of Medicine discovered a set of learning and memory mutations. And they named them things like rutabaga and amnesiac, a number of clever things. Turns out one of the critical learning and memory mutations, this is a mutation that they learned fairly well, but they tended to forget fairly quickly. And then there's some that don't learn so well, but those that did learn it, they remember it about the same 3 days. Well, one of the learning and memory mutations, I'm pretty sure, is the guys that tended to forget. When the mutation is identified, it's like, oh, this is significant. We now have a gene involved in learning and memory. And it takes a couple of years. They actually clone the gene, sequence it, and what is it? It's adenylyl cyclase. It is the same adenylyl cyclase that's in every single cell. It's like, whoa. What we got here is a cell signaling event that's happening in your neurons that, with specific responses, you're inducing cyclic AMP, and you're having an immediate response, but you're also going to have a long-term response, which we'll be getting into on Wednesday. And so, there you go. Adenylyl cyclase, this is the effector. The second message is cyclic AMP. Thank you very much. Exams are still in the process, whatever. Oh, lab tomorrow, we're going to start kind of our final lab thing where at the class I'm going to call the Moequa Packing House, which is what I've been going to, because I don't think I'm doing, because I used to go to the place in Bloomington. And I'm hoping I'm going to get some kind of odd cow part, like a knee. Something that's got a lot of different tissues in it, and we're going to extract proteins from these different tissue types and then look at them by protein electrophoresis next week. So I'll probably do a little bit of a video introduction on that, but we'll see how that goes. And there you go. So we have some stuff that's happening in the lab this week. That's it. All right. Bye.